Okay guys, let's make this presentation little bit more exciting, and for this we can take a help of a story. So do you all know guys that in my childhood I want to become an army officer, and I think that becoming an army officer is the dream of every third person in his childhood. And why not? They have their own communication system, own defensive system, their various rankings like soldiers, generals, colonels, etc. But you guys literally wonder when I say you that similar system also exists in our body. Amazing, है ना? Good morning, all of you. I am present here, myself Aloksa, to give you presentation on my topic immunoglobulin. Yes. So before starting my presentation, I want to ask you that are you ready, guys? Okay. So let's start with immunoglobulin. Immunoglobulin as terms itself suggests that it is made up of two little terms like immuno and globulin. Immuno means immunity and globular means globular protein. Globular proteins which provide immunity. Okay. So these are also known as glycoproteins, and these are Y-shaped in structure. They have two heavy chains and two light chains. They are made up of carbohydrates and proteins. They are present in serum. Okay, and if I talk about their structures, I show you the structure of this immunoglobulin. This is the structure of immunoglobulin. There are two heavy chains, two light chains. They are bonded with disulfide bonds, and these are the epitope region, which are variable region. These are the constant region, and in center there are carbohydrate. Okay, so special feature of this immunoglobulin is that after analyzing the foreign antigens, our antibodies can change its structure. It is like lock and key system. In that we know that various types of locks needs different kinds of keys. If correct lock have correct keys, then that lock get open, वो टूट जाता है ताला या फिर खुल जाता है जब उसको सही चाबी मिलती है. In this key there are two parts. One is where you hold it and one is which part you going to insert in the lock. Okay, so this inserting part is बदलता रहता है basically according to the foreign एंटीजन. ठीक है, आपका फॉरेन एंटीजन यहाँ पे ताला है और जो आपका ये जो एंटीबॉडीज है वो चाबी है यहाँ पे. ओके, सो जो ये कंस्टेंट रीजन जो मैंने बताया जहाँ पे वो होल्डिंग पॉइंट है विच इज यूज्ड फॉर ग्रिपिंग पर्पस इन प्रैक्टिकल लाइफ इन प्रैक्टिकल कीज. ओके, सो दिस ऑन दिस बेसिस बेसिकली यहाँ पर 5 टाइप्स ऑफ इम्यूनोग्लोबुलिन होते हैं. ओके, so these are IgG, IgA, IgM, IgD and IgE. I show you on my presentation slide. This is the type of immunoglobulin. There are the 5 types of immunoglobulin, and this is also known as other names also. That IgG also known as gamma, IgA is also known as alpha, IgM is also known as mu, IgD is also known as delta, and IgE also known as epsilon. If I talk about their concentration in the blood, so this as follows: that IgG is have more concentration than IgA, then IgM, then IgD, then IgE. Okay, so this is the concentration in the blood. Now let's take a look on the function one by one. These are the functions of immunoglobulin. First we take IgG. Okay, so IgG is monomeric immunoglobulin. Basically what is monomeric? It is one Y-shaped immunoglobulin. One antibody can bind with two antigens. Okay, so this IgG is basically responsible for protecting our body fluids. Okay, this is the only antibodies which can pass the placenta. It is responsible for giving us immunity against chronic diseases. What are the chronic diseases? Chronic diseases are those diseases which continues in our body longer than 1 week. Okay, And their main principle of IgG is phagocytosis. How they perform this phagocytosis? Immune cells basically have the receptors. This receptor is nothing but your immunoglobulin. Okay, so this immunoglobulin is basically responsible for detecting that foreign antigens, and this immune cell is responsible for destruction of these foreign antigens. But they have this immune cell don't have any detecting feature, and this receptor don't have any destruction feature. So they work in collectively to show their response and counter the foreign antigens. Okay, so this is how phagocytosis work in case of IgG. If I talk about their types, there are four types of IgG immunoglobulin. There are Ig1, Ig2, Ig3, Ig4. Okay, so Ig1, 2, and 4 basically have 24 days half life, and Ig3 has 8 days half life. Okay. Now, if I talk about our next immunoglobulin, which is IgM, this IgM is pentameric in nature. Our no any other immunoglobulin have pentameric. What I mean with the pentameric? Pentameric means basically they have five Y-shaped their immunoglobulins connected with J chain, and that can bind with 10 antigens, because one can bind with 2, so 5 can bind with 10. Okay, so if I talk about more about IgM, then this is basically showing primary response or secondary response. What are primary secondary responses? Primary response when foreign partisan came in our body to harm us, so our immune cells show primary response and also save some second memory cells for future encounter. So whenever this same antigen came in future again, then our saved memory cells also work and give it a counter very fastly and effectively than the earlier one. Okay, so this is how primary secondary response. IgM is the first antibodies which made in newborn babies. Okay, after 6 months, and IgM basically this is all about IgM, I think. And now we have to go on next our immunoglobulin, which is IgA. IgA is basically dimeric. Okay, it means it have two Y-shaped immunoglobulin. Okay, they have also secretory component, also a J chain. Okay, it can bind with four antigens. Okay, so IgA basically also found in colostrum of the mother milk. Basically, newborn baby don't have any antibodies. Their IgM synthesis after 6 months. So this IgA play a crucial role in the immunity or to in newborn baby's case. Okay, this IgA is responsible for counting the diseases like influenza, Vibrio, and various diseases like polio. Okay, so these are the diseases, and IgA is basically more. If I told that IgA is I think this is all about IgA. Next is IgD. IgD's biological activities are less known. Scientists and researchers are still discovering their new discoveries about them to know their more biological activities. And till date, we know that IgD is monomeric in nature. Next is IgE. IgE is basically what IgE is also monomeric in nature, and their receptors are present on mast cell, eosinophil, basophil. Basically, their predominant role in your allergic and hypersensitive reactions. Okay, so this is all about the functions of immunoglobulin. I tell you IgG's function, IgA's function of Ig MDE. Okay, there are five immunoglobulin, and I also tell you the classification of this immunoglobulin, and also we see the deep knowledge of structure. And this is all about my presentation. I hope that you all guys understand my every topic and also enjoy my presentation, and I am very thankful to all of you to pay me attention throughout my presentation. Okay, but still if you have any doubt, you can ask to me. I am listening to you. Anybody? No, it's okay, no problem. At last, I want to say that do not let your fear to take over. Face it head on. You know that you are stronger than it is. Thank you.